My name is Ryo. I'm a PhD student at UCLA, co-advised by Dr. Shao and Dr. Zaylin. I want to thank organizers for giving me an opportunity to present my work. Today, I'm going to be presenting my project, where I developed a tool to detect double-stranded RNA using long-read RNA sequencing. Double-stranded RNAs are RNAs with inter- or intramolecular double-strand bindings, and it's a known feature of viral RNAs. To protect itself from viral infections, viral double-stranded RNAs are sensed by immune-activating sensory proteins to activate immune response. However, RNA transcribed endogenously from your own genome can also form double strands and falsely activate immune response. To prevent falsely activated immune response, adenosine deaminase acting on RNA, also known as ADAR, binds to endogenous double-stranded RNA to edit adenosine to inosine, deforming the base pairing of double strands. Edited double-stranded RNAs are likely to be not sensed by immune sensory proteins because of altered structures. And accumulating evidence suggests that the dysregulation of this process is associated with diseases such as cancer, inflammatory diseases, and autoimmune diseases. Therefore, identifying immunogenic double-stranded RNAs is an important task to advance understanding of these diseases. Common way to identify double-stranded RNA includes the usage of editing enriched regions identified from RNA-seq data. Because ADAR protein is a known double-stranded RNA binding proteins, double-stranded RNAs that are heavily edited are likely to be immunogenic. However, when ADAR activity is compromised in certain conditions, such as in diseases that I mentioned, using editing enrichment for double-stranded RNA identification might miss some important functional immunogenic double-stranded RNA. So given these challenges in current double-stranded RNA detection, our goal is to develop a new method to identify double-stranded RNA without the need of clonifying A2I editing. Yes, so but how do we identify double-stranded RNA without RNA editing? So previous studies have reported the strong double-stranded regions of RNA might get skipped by reverse transcriptase, as they cannot unfold the strong double-stranded structure of RNA. And because this skipping is not a well-regulated process and just happens because of the structure, might be different from skipping that happens through regulatory process that we always see, such as splicing. So we could potentially utilize this to detect double-stranded RNA region. So let's look at some examples. Here I am showing two examples of skipping that is happening in canonical splicing and double-stranded RNA region, respectively. Each row represents reads, and the gray part represents regions covered by reads, and blue part represents skipped regions. So you can see that the canonical splicing has skipping start and end site exactly the same across all reads. Since splicing is a regulated process, there's no variability in skipping across reads. If you closely at skipping in double-stranded RNA region, you can tell that starting and ending are all relatively random. Single-stranded region is transcribed consistently across reads. However, regions that are structured with double strands, shown in red box, have skipping that are not consistent across reads. As I previously mentioned, this is because reverse transcriptase not being able to unfold the structure of RNA when creating cDNA template. And because this is not a regulated process and occurs randomly, skipping patterns are stochastic. So inspired by this observation, we further explored to see if we can leverage this artifact of long-read RNA sequencing to detect double-stranded RNA independent of editing. So we developed dsRID, short for double-stranded RNA identifier. Using the mapped reads from long-read RNA-seq, we extract features that are related to stochastic skipping, such as length of skipping, frequency of skipping, standard deviation of skipping sites, GC content of skipping, and etc. Using these features, we train random forest classifier to identify potential double-stranded RNA region. For training purposes, here positive sets we used are structurally validated, editing enriched double-stranded RNA, and negative sets are regions randomly sampled outside the positive regions. After the model is trained and predictions are made, we filter those regions by curating secondary structure using RNA-fold to ensure long stretch of double strands. In this project, we tested our method on 11 different samples. All samples are sequenced using PacBio long-read RNA sequencing protocol. One sample from the Alzheimer's disease patient brain region generated Pacific Biosciences. One GM12878 cell line from ENCODE, and 580 and control brain region sample also from ENCODE consortium. A plot up on the right showing that the number of region in the training set on y-axis and sequencing depth on the x-axis, showing that with more coverage, there are more regions to train the model with. We used the model trained on the most deeply sequenced PacBio Alzheimer's disease data and tested their performance across all different samples. Here we show the receiver operator curve or method across different cohorts, and we see consistently high performance of dsRID across different data sets. And plots on the right shows the area under the curve score for each sample, and we see average AUC score of 0.94 to 0.95. After seeing that our model performance is high, we went ahead and tried to characterize editing levels of double-stranded RNAs that we identified using previously curated editing sites. We can see that as expected, novel double-stranded RNAs that we identified are edited similar level compared to non-ER-based double-stranded RNAs, and higher editing levels compared to structured RNA, which are the negative control regions, also indicate that our double-stranded RNAs are confidently forming double strands. And here we tested whether there's a good overlap between experimentally validated double-stranded RNA with their prediction. And J2F-ClipSeq is the experimental protocol to pull down double-stranded RNA using antibody and is considered gold standard in double-stranded RNA detection, but with very limited sensitivity. And compared with the J2F-ClipSeq data on HeLa cells, our dsRID prediction have much higher fold enrichment compared to traditional ER-based approach. After seeing that our method is validated, we proceeded to characterize double-stranded RNA identified in Alzheimer's disease sample and control samples. We found that a lot of double-stranded RNAs are shared between AD and control. However, we found some specific double-stranded RNAs that only appear in AD samples and control samples. We see that the double-stranded RNAs that are specific to AD samples is less edited, shown on the plot on the left, meaning the hypoediting of these double-stranded RNAs potentially contributing to false activation of immune response in Alzheimer's disease. We also observed that the total expression of double-stranded RNA is higher in AD samples compared to control. This might also be explaining that AD samples tend to have more double-stranded RNAs that are less edited, that are immune stimuli within these cells. In conclusion, we show that dsRID performs robustly well across several cohorts. dsRID predictions are validated through A2I editing levels and through external experimental annotation. And comparison between AD and control using dsRID shows potentially distinct dsRNA profiles, and this project's being published. So if you're interested in using this tool, please look up my paper, scan my QR code. And I just wanted to acknowledge my PI, Grace Shao, and all collaborators, and I'm happy to take any questions.
Any questions for you?
How are you distinguishing, say, different isoforms, or in particular those induced by cryptic splicing? And relatedly, what would a cryptic variant look like in this sort of output versus double-stranded RNA skipping?
That's a good question. So I guess, yeah, cryptic splicing, it depends on basically how you would see, right? Basically, in this study, we kind of know that the splicing patterns that are not on the basically splicing donor acceptor sites are like, we consider them double strand, or there's like some structure that induces skipping. So yeah, I guess it's hard to tell if without like annotated basically splice sites.
Thanks.
Yeah.
Hello. Hi, great talk. I have a question for you. So one question that we've had in the AD field is what's the main source of the dsRNA? Is it from mitochondrial sources, or is it from transposable elements being degraded?
Mm-hmm.
And the sequences would differ, but you also have long read, so you could also measure potentially retrotransposons and transposable element copies. I was wondering if you've looked into that. And then the second question is, do you have any sequence-based inference of what cell type they may be coming from?
Yeah, so to answer the first question, we analyzed what kind of basically the ratios of the dsRID double-stranded RNAs. So obviously it's enriched in sign elements because the ALUs are usually edited, and that's why we consider immunogenic double-stranded RNAs. But we see some enrichment in line elements, simple repeats, and so on. But I didn't like specifically do like a comparative analysis between AD and control. And in terms of cell types, so as you can see, our data is pretty limited. We only did it in bulk samples with like 4 or 5 samples, and that was the comparison. So if the single cell long read sequencing with like AD, then we can like actually dig deep into what cell types are contributing to double-stranded RNAs.